Welcome everyone to this edition of Precision Nanosystems Tea Time webinar series. Thanks all very much for attending. On this edition we have Dr. Samir Cherkaoui from Bracco, and Samir will be discussing microfluidic-controlled manufacture of monodisperse perfluorocarbon nanodroplets for ultrasound imaging and therapy. So just before we get started, just to introduce the Precision Nanosystems team. So if you have any questions either about today's session, you can contact Samir at Bracco directly or contact one of us. Or if you have any other questions, please feel free to reach out. So you have myself and Richard, who are based out of the UK, and you have Jürgen and Martin, who are based out of Germany. So at this point, I'd like to hand it over to Samir. Thanks so much, Samir, for presenting, and over to you.
Okay, here we are. So I'm pleased to be digitally with you for this Tea Time webinar. First of all, I would like to thank the Precision Nanosystem team for giving me this opportunity to share with you some of our results using lab-on-a-chip or microfluidic platforms for the manufacturing of monodisperse bubbles and nanodroplets. So I will start my presentation with a brief introduction on Bracco Group and its main activities. Then I will provide an overview on the use of two microfluidic platforms to prepare monodisperse bubbles and monodisperse PFC or perfluorocarbon nanodroplets for ultrasound biomedical applications. And then I will conclude with some general conclusion. So Bracco Group is an international pharma company created in 1927 and headquartered in Milan, Italy. With more than 3,000 employees, the company is acting globally with different R&D sites, mainly in Europe and the US. We have also different manufacturing sites worldwide, but also medical sales and marketing facilities. The Bracco Group is composed of three different business units, the largest one being Bracco Imaging. In fact, Bracco is an international leader in diagnostic imaging. Then ACIS Medical, this is leading the development of smart systems and medical device for the administration of contrast agent. Finally, CDI, this is an Italian diagnostic center in Milan, and this is a kind of polyclinic facility providing services in terms of diagnosis and rehabilitation. Concerning the Bracco Imaging is involved in different imaging modalities such as X-ray CT, nuclear medicine, MRI, and contrast-enhanced ultrasound. Bracco, for each modality, we are providing different contrast agent with the aim to improve the imaging procedures, making easier the detection and characterization of various pathology. In the Geneva site, we are mainly focused on the design and manufacturing of microbubbles with our well-known Lumazon in the US and Sonoview in the rest of the world. So, for some of you who knows a bit about ultrasound contrast agent, they are mainly gas encapsulated microbubbles, and they are used to improve the echogenicity of the blood. In the case of Sonoview, a contrast agent developed and commercialized by Bracco, is it based on the use of an inert and low solubility SF6 or sulfur hexafluoride gas stabilized by a lipid monolayer. In this case, we are talking about micron-sized bubbles with smaller diameters, even smaller than red blood cells that can pass smaller capillary. Microbubbles, when subjected to ultrasound, they will cavitate. They starting from linear cavitation at low acoustic pressure to nonlinear cavitation to nonlinear oscillation at medium acoustic pressure. So thanks to the high compressibility and this nonlinear oscillation of the bubbles, they can be detected using clinical ultrasound machines. So we are using this feature, these microbubble features, because they allow us to nicely visualize different organs, especially lesion in the case of the organs. In this video, you can see how we can use the microbubble in the contrast mode to better image a lesion, in this case, a liver, and you can see how the microbubble can be nicely visualized. Most of ultrasound contrast agents are polydisperse suspension with the size diameter ranging from 1 to 10 micron. So tightening the size distribution of the microbubbles is nowadays considered as a way to unlock the full potential of microbubble for ultrasound biomedical application. As you know, as shown before, microbubble are acoustic resonators with a characteristic frequency that is inversely proportional to the size of microbubbles. At resonance, of course, both the amplitude of oscillation, but also the nonlinear echo are at maximum. So, in contrast to polydisperse microbubbles, monodisperse bubble will react in the same manner when subjected to ultrasound wave. In other words, every bubble will count in the case of monodisperse population. So we really believe that using this monodisperse bubble will further improve the sensitivity of diagnostic imaging, but also theranostic applications. So at Bracco, we are interested on the use of microfluidic technology to produce these mono-size bubbles. In this case, we are using what we call the flow focusing device. Briefly, a gas thread is focused by perpendicular lipid suspension through a narrow orifice where the gas thread pinches off to release monodisperse bubbles. The accurate control of the gas pressure, the flow rate, and the orifice dimension will allow us to produce highly calibrated microbubbles. So using this approach, we are now able to produce highly micron size bubbles with different size, micron size bubbles with a highly monodisperse and controllable diameter at a very high production rate. Beside the device sitting, the coalescence and stability of this microbubble is key during the process. In fact, the coalescence of freshly formed monodisperse bubble may lead to decrease or to lower concentration of the microbubbles, but also to polydispersity, leading to non-uniform acoustic response. In this case, you can see by just replacing PEG 2000 by PEG 5000 in the formulation, we greatly impact the coalescence phenomenon. We further characterize the coalescence probability and the long-term stability of the bubbles as a function of different formulation parameters and more particularly the temperature. And it comes out that by increasing the temperature, we drastically decrease the coalescence probability. So working at high temperature with a common DSPC PA-PEG 5000 formulation, we are able to produce monosize bubbles, monosize and stable bubbles over several days. Now let's move to another topic dealing with acoustically activated or phase change nanodroplet. These agents are rapidly evolving in both diagnostic and therapeutic delivery. Except the core, which is generally formed by liquid perfluorocarbon, this nanodroplet display the same structure as commercially available microbubbles. So thanks to the acoustic droplet vaporization process, this encapsulated nanodroplet will be converted to gas microbubbles upon exposure to acoustic energy beyond an ADV threshold. Of course, smaller size nanodroplet will require higher energy. Besides the low ecogenicity of the nanodroplet, thanks to the nanometric size of the nanodroplet, they will allow increase in vivo circulation and also deep penetration in the tissue. So this nanodroplet are also ultrasonically stable and can be acoustically vaporized. So nowadays, PFC nanodroplet present several interests in different therapy area. For example, in drug and gene delivery, in fact, they are now want to promote sonopermeabilization, to also safely promote BBB disruption, and can be also used as theranostic. They can also use in therapy for thermal ablation, sonothrombolysis, can be used also for multimodal imaging. In particular, since the nanodroplet can be activated and deactivated on demand, they are considered as an interesting agent for super resolution imaging to super resolve microvasculature. Finally, nanodroplet can also be used to perform passive and active targeting. So how to produce the nanodroplet? Of course, most of you know about conventional procedures such as sonication. It's an easy method, really simple to use, cost effective. But this procedure requires higher energy and generally lead to low production volume. We can also use microfluidizer in order to reduce the particle size into the nanoscale level. And this allows us to produce decent nanodroplet material for R&D, for exploratory R&D up to pilot batches. However, once again, this method requires high pressure and high shear rate with the risk to deteriorate the encapsulating material. In addition, in the case of nanodroplet, the increase in the temperature may result in liquid PFC evaporation. The last procedure is dealing with microbubble condensation. In fact, this is called, I would say, a top-down approach, starting from commercially available microbubbles with highly volatile gases, and by appropriate control of pressure and temperature, we can easily condensate this microbubble to generate nanodroplets. However, the generated nanodroplets are generally polydisperse and with a rather low concentration. So, in general, all this conventional method suffers from some drawbacks in terms of uncontrolled size and consequently non-uniform acoustic response, polydispersed population, and finally, the risk of degradation of the nanodroplet itself or the encapsulating therapeutics. So there is a need to find the alternative method in order to better control the size and polydispersity of the nanodroplet. In our case, we are targeting a size between 200 and 400 nanometer with a PDI below 0.2. So naturally, we move toward microfluidic technology. So most of you know about the microfluidic platforms from Precision Nanosystem. This is a kind of Swiss knife allowing us to produce different nanomedicine, including the well-known liposome, LNPs, emulsion nanoparticle, and so on. These platforms offer several advantages in terms of robustness, batch-to-batch consistency, and more importantly, the scalability. So how we can use this platform to produce our PFC nanodroplets? In our investigation, we use the nanoassembler system with a microfluidic cartridge based on the staggered herringbone. As for other formulation, the aqueous phase is mixed with organic phase composed of perfluorocarbon and the encapsulating agent, which may be a lipid, fluorinated surfactant, or a polymer. So the two streams are well mixed thanks to this architecture of the mixer, and they are mixed in a controlled manner to generate the nanodroplet that can be collected at the outlet of the cartridge. During our investigation, we evaluate different parameters, including the shell, namely phospholipid, fluorinated surfactant, and PLGA polymer. Of course, we have also assessed the effect of different liquid perfluorocarbon with different boiling point. And once again, we evaluate also the effect of process parameters on the nanodroplet characteristics. So the quality attributes of this nanodroplet are key. That's why all the nanodroplets were systematically characterized in terms of size, polydispersity, and zeta potential, but also the stability was systematically evaluated. This is a common approach during the optimization of nanomedicine using the nanoassembler system. Of course, we can adjust the TFR, the total flow rate, and the flow rate ratio in order to fine-tune the formulation in terms of size and PDI. As expected, of course, increasing the shear stress will decrease the nanodroplet size. We have also evaluated the effect of flow rate ratio, and it must be noted that during our investigation, it comes out that depending on the encapsulating agent, the behavior of FRR effect was quite different. Finally, we have also evaluated the effect of the perfluorocarbon concentration, as shown here. Without perfluoropentane, liposomal formulation were generated, and by increasing the PFC concentration, result in larger nanodroplet with heterogeneous size distribution. So all these parameters can be used to fine-tune the formulation during the manufacturing process. So our first investigation were carried out using lipidic material as a shell. And as you can see here, by using DPPC and DSP PEG mixture, we have been able to generate nanodroplet with a size around 300 nanometer with a decent concentration, but the stability was not sufficient. Then we move to fluorinated surfactant. We use a simple zonil as a surfactant, and by setting the microfluidic process settings, we have been able to generate, once again, monodisperse suspension with a size around 300 nanometer, and the stability was found quite good. Finally, we used also polymeric material. In this case, the PLGA was solubilized in acetonitrile, and polyvinyl alcohol was added in the aqueous phase in order to better stabilize our nanodroplet. Here again, we achieve monodisperse population with very good stability. So now we developed also, we implemented the homemade acoustic setup in order to characterize our nanodroplet. Briefly, the diluted nanodroplet were circulated in a tube at 37 degrees and were exposed to a single element transducer at 6 megahertz, and which allow nanodroplet vaporization. In parallel, a linear probe was used just above the tube in order to collect videos in the B-mode imaging. Thanks to the use of our ViewBox quantification software, echo powers value were derived. During this investigation, the acoustic pressure was increased every 10 seconds in order to observe until the occurrence of the vaporization of the nanodroplet. So, as illustrated in this video, by increasing the acoustic pressure, when we reach the ADV threshold, our nanodroplets are vaporized to generate echogenic microbubbles. So The echo power values were reported as a function of acoustic pressure, leading to a sigmoidal curve, and the ADV threshold can be easily determined. In the case of Zonil nanoformulation with the size around 300 nanometer, we have been able to determine the ADV threshold around 4.4. The similar values were obtained in the case of lipid-shelled nanodroplets. However, in the case of hard-shell nanodroplet with PLGA polymer, it was not possible to determine the ADV threshold under the present acoustic conditions. So we further investigate the influence of the nanodroplet size on the ADV threshold. And as already reported by other groups, the ADV threshold was inversely proportional to nanodroplet size. The nanodroplet concentration is also key during for in vivo efficacy and safety. So we assess also the effect of ADV threshold, the concentration on the ADV threshold, and it comes out that the ADV threshold is really slightly affected by the concentration in the studied concentration range. Another application of the microfluidic device was used to produce nanodroplet with the perfluorocarbon mixture consisting of perfluoropentane and perfluorohexane with boiling point around 29 and 57 degrees respectively. So in this case, the mixture was introduced in the organic phase, and once again, a mix was used between the aqueous and organic phase to generate the nanodroplets. So as shown on the table, by increasing the perfluorohexane content in the core, the size was slightly affected by the composition of the perfluorocarbon. Whereas the ADV threshold was increased with the perfluorohexane content, apparently using perfluoropentane used as a trigger to allow the vaporization of the perfluorohexane. Finally, the last slide is about the scalability. As you know, Precision Nanosystem has developed a new technology based on this toroidal mixer, allowing large-scale production of nanomedicine. So we had a chance to compare the conventional staggered herringbone microfluidic mixer with this new technology. This investigation was performed using all our nanodroplet consisting of lipids, polymers, and fluorinated surfactants. As we can see, whatever the platform used, benchtop, Ignite Classic, or Ignite NxGen, we have been able to achieve similar nanodroplets in terms of size and polydispersity. This further demonstrates the seamless transfer from lab scale to large scale for this nanomedicine. So this will allow us to anticipate future GMP manufacturing to support our preclinical and clinical needs for ultrasound applications. So we come to the conclusion, we really believe that microfluidic technology may play a role in the preparation of nanomedicine, in our case, in micro and nanoparticles for ultrasound diagnostic and therapeutic application. Using flow focusing device, we have been able to prepare monodisperse microbubbles with the uniform acoustic response, and also monodisperse nanodroplet. In the case of nanodroplets, the challenge is really to find a compromise between the ease of vaporization and the chemical and thermal stability of the nanodroplet. Finally, now we have in hand an efficient tool to better characterize this vaporization of nanodroplet, leading to echogenic microbubbles. Last but not least, I would like to acknowledge all my colleagues from Braco, and also our collaborator from Twente University in the Netherlands, but also from Avignon University in France. And thanks to all of you for listening. Of course, this is a tea time webinar, but we can move to some champagne or Prosecco to enjoy the taste of the microbubbles. Thank you for listening.
Well, thank you very much, Samir. That was a really interesting application, really interesting talk. I'll probably have the champagne or the Prosecco ready at the moment. I just have a blackcurrant juice at the moment, but maybe later. So we're at the top of the hour now. So unfortunately, I'm going to have to say thank you to everyone. If anyone has any more questions we haven't been able to answer in the live session, we will follow up by email and get those answered for you. But all that remains for me to say is, firstly, thank you very much to our speaker, Dr. Samir Cherkaoui. Really appreciated that, very interesting. And thank you all, everyone, for attending the seminar again. And we look forward to seeing you all again for the next Tea Time webinar in 2 weeks. Thank you, everyone.
Thank you. Bye-bye.
Bye-bye.